When we talk about genetic engineering today, genetic engineering is this idea that we can use modern DNA technology to combine DNA from different organisms together. By using molecular techniques, not literal tweezers, we can add or remove specific genes in order to create genetically modified organisms. This is something that we have been doing in a sort of primitive way for tens of thousands of years by selectively breeding certain organisms who have traits that we like. We can create organisms that have a mix of the desired characteristics. Height, in this example, or number of seeds, or sweetness, or lack of aggression, or whatever it may be in the plants and animals that we depend on. We've done this with many of our food crops like watermelon, like eggplant, like carrots, like bananas, and even with dogs. That the dogs that we have around us today all descended from wolves who associated with humans and whose breeding we selectively targeted and controlled in order to create the dog breeds that we have around us today that have many of the special characteristics that we like. In the 60s and 70s, we started doing this in a more targeted way by manipulating DNA directly, first in single celled organisms, then in more complex organisms like mice, which have now become a staple of genetics research. The first food that was genetically modified was the Flavor Saver tomatoes, which had DNA technologies that were added in order to remove the activity of an enzyme that caused rotting. So this tomato stayed fresh for longer. We can now do this with many different species of plants and animals to make them grow faster, to be resistant to disease, to produce more food for the amount of food that we give them. There are still people in society who are very afraid of genetically modified foods. There have been many studies. Every study shows that there are basically no ill effects from eating DNA that's been modified because your body treats it all as food and destroys it all down to its molecular components. There are no real changes to the food directly either, but there are still people who believe this to be dangerous in some way, even though most of the food that we eat, especially packaged and processed food like cereals or chips, contain genetically modified organisms in them. And we've been eating them for decades with no real safety issues. There will still be skeptics, and this is this anti-science thing we see with the pandemic as well. Right now, instead of killing animals to extract life-saving therapies such as growth hormones or insulin, we rely on genetically modified organisms, fungi, and bacteria to make these products in the lab by inserting human DNA into these organisms and helping them to create these life saving molecules in large factory like reactors. Right now and into the future, a new technology called CRISPR, which uses this tiny molecular machine called Cas9 to make very targeted edits to DNA, is going to be very powerful to help us create new life saving therapies and potentially change the nature of society as we know it today. CRISPR can be used to modify every single type of organism, single cell to more complex, plant and animal too. I do want to emphasize this is not currently feasible. This is 10, 20, 50 years into the future. We could potentially edit human DNA to change our immune system to be resistant to disease, to tweak embryos to have a mix of characteristics that we like, and even reverse some of the aging that can come from DNA damage. I hope this is some food for thought as well. That this kind of therapy is currently banned. Research into this is currently outlawed by most countries around the world. A scientist, Professor Hay, was jailed in 2018 for editing the DNA of two embryos using CRISPR to make them resistant to HIV. This therapy is banned because of the unstudied nature and because of potential ill effects on the embryos. Please respond to these questions on Canvas.